Welcome everybody. I can see a lot of smiling faces out there, but I'm also pretty certain that one of the reasons we're all here is because we've been touched by this hideous disease called cancer. And what you will learn today, and what we've been discovering a lot already during the conference, which has been going today and will go tomorrow and Saturday, is that there's a revolution happening right before our eyes, and that's where we're training the immune system, which is designed to get rid of bacteria and viruses and infections. We can now use our technology smarts to engineer the immune system to attack cancer. And so what we're wanting to do is convert those words of fear when you hear, hopefully no one in this room will ever hear the words "you have cancer," but let's try and replace the words of fear with one of hope. And so what we're going to do in this forum is bring you up to speed with some international experts that will tell you about the basics of immunotherapy and some of the outcomes. And we're particularly indebted to Jared Rufford, who will join us in around about 6 o'clock, and he will tell his personal experience with Grant MacArthur as his doctor. So the event tonight is a joint venture between the Eradicate Cancer Conference, which we've been convening, together with the Melbourne Immunotherapy Network, and we're very pleased to have the chairman of that network, Dale Godfrey from the University of Melbourne. David.
Thanks very much, Susan. Peter, Richard. Thanks everybody for coming along here. The Melbourne Immunotherapy Network was formed in 2016, and the idea is to bring together clinicians and lab-based researchers together at a few meetings each year, with the idea that these people they're both interested in immunotherapy, but they don't often work in the same circles, and so getting these people together will give us newer, better ideas to improve immunotherapy and ultimately lead to better and safer forms of this remarkable new way of treating cancer and other diseases. So the Melbourne Immunotherapy Network is a collection of entities, including the University of Melbourne and the Doherty Institute, Monash University, the Peter MacCallum Cancer Centre, WEHI, the Royal Melbourne Hospital, the Olivia Newton-John Cancer Research Institute, St Vincent's Institute and Federation University, and other players in the Melbourne and surrounding area. We have a series of activities each year, including annual symposiums. We're planning a winter retreat this year, and public forums like this one, and this is the second one we've had, and I'm really pleased to see how many people have come along to hear a bit more about immunotherapy. And I think, I'm really encouraged that I think this network's going to make a big difference for the type and the quality of immunotherapy research that's going on in Melbourne. And I really want to say again, thank you. We are accepting donations. If you'd like to provide a contribution, there'll be people handing out flyers at the door to give more information about that. So thanks again, and welcome.
Thanks very much, Dale. And it also gives me a great pleasure to introduce the two conveners for the session this evening. The first is Professor Grant MacArthur, who's the Executive Director of the Victorian Comprehensive Cancer Centre, and he's also a global expert in this field of immunotherapy. Grant. And our Grant's co-convener is a very close friend and colleague of mine, Professor Alan Trounson. Many of you will know him through his world-shattering work in vitro fertilisation and in latter days in the world of embryonic stem cells and then stem cells. And he is actually now one of the global leaders as well in this field of immunotherapy. And I'd like to introduce Alan, who will introduce our guest of honour.
Thanks, Richard. I was just waiting for our guest of honour downstairs, and there's a Arnold Schwarzenegger group down there, and they kept confusing me with Arnold, and I said, I'm here waiting for somebody really important. And he said, well, you must be Arnold's cousin. So, welcome everybody. I think there are some really key moments in time, and this is one of them, I think, and it's really just been brought together with this conference and this public event. We had the Minister of Health, the Federal Minister of Health here this morning, And he was very supportive of us actually doing something together as an immune therapy group. And the kind of work that Grant and all the others have been doing here in Melbourne as a sort of really leading the world in immunotherapies is something incredibly impressive. And I think we can do something very special here. So I'm actually on a committee with Frank Maguire, and we share on this committee, and there's a number of us on the committee. And I think Victoria wants to do something special in this kind of space as well. So I think this might be one of those moments, and I'm hoping that Frank might open up this as an opportunity for us in what he's going to speak to us about. He's going to open this public session. He's Parliamentary Secretary for Medical Research, and that's the first time we've had one in medical research. He's also Parliamentary Secretary for Small Business and Innovation, and together, on this SMART committee where the Health Minister is involved as well, this really means that we're able to sort of input into the Victorian government in a way where we can sort of provide them with opportunities. This might be one, but there are many others in the medical area that will really make a difference, and we want to make a difference for all the people. So Frank is incredibly well known. He won Walkleys and all sorts of things as a journalist. He's a well-known businessman. He's really been been one of the key parliamentary people that's really engaged with medical research and taken the message to the Victorian government. He is able to really get our message across to the people who are really making policy and creating opportunities here in Victoria. And I'm so pleased he's agreed to come and open this session. And I think the session is for all of you to see what might be able to occur as a result of these incredible developments in immunotherapy, and they really are incredible. And I think you can imagine that maybe in the future, in 10 or 15 or 20 years' time, the burden of cancer is not going to be like it was today. And we're on our way, I think, to eradicating cervical cancer. I would hope that we'll be on our way to getting rid of a whole lot of others, at least reducing the burden. I think we've done incredibly well at HIV. I think we're able to do that, and I think the scientists and the people that you hear today will talk about that. But introducing Frank Maguire, thank you very much, Frank, for coming, and we look forward to hearing some of your views with respect to this area. Thanks.
He's way too brilliant to be confused with Arnold Schwarzenegger, isn't he? Come on, let's call it out. No, a wonderful man. Now, thank you for that very gracious introduction. I'm delighted to be here. It's always an honour. It's very humbling to be with so many of our absolute leaders. And it's not just out of Melbourne or out of Australia, that's at international standard. And for what they do, and I always say to the sector, it's a wonderful group of people to be involved with, because they're culturalists, they're not monetarists. They do things because it gives them meaning. And what's the meaning? They want to take fear and turn it into hope. They want to actually collaborate to get something done that's meaningful, that can save lives. So I have great admiration for the distinguished people who are here today. And if you're in this room, you're distinguished. What about a round of applause for our distinguished guests here? Come on, give yourselves a round of applause. And I would like to specifically recognise Alan for all the work that he does, for Sharon Lewin as well from the Doherty Institute, and we sit on the SMART panel, the Science, Medical Research and Technology panel. And what we're trying to do with that, this was an initiative I put up, and Premier Dan Andrews, former Health Minister, has a great interest and insight into all of these areas. And Jill Hennessy, as the Health Minister, I'm delighted to work closely with them in a collaborative way to try and get better results. So they saw this proposition that how do we actually get the idea of getting capital to the idea so that we then invest in what the breakthroughs are, so we take them from bench tops into businesses, and then we actually look at, what are the cures that we can have, and I'm trying to drive that we get the value out of our intellectual property here and we manufacture here whatever the remedies and the solutions are. So that's one of the things that the SMART panel does. One of the other things is that we're looking at with the Medical Research Future Fund. So this is a $20 billion investment over 20 years. And what are the big propositions that we want to tackle? So Victoria does lead in this. It's not a matter of a parochial view, it's a matter of fact. That basically funding from the NH and MRC, Victoria regularly gets more than 40% on merit, on performance, and on the institutional excellence that we have. So the lion's share goes to the lion, and my view is that we should lead like the lion. So that's what we're trying to do here in Victoria. So we're really great to see so many people here with such a commitment and to work out how we can collaborate further. I'd like to also acknowledge Cassie and Yi from Memorial Sloan Kettering Cancer Centre, Grant MacArthur, of course, the co-chair of the Melanoma and Skin Service and the head of Molecular Oncology Laboratory and head also of Translational Research Laboratory at the Peter MacCallum Cancer Centre, Rebecca Delconte, Miles Prince, Director from the Centre for Blood Cell Therapies, Director of Molecular Oncology and Cancer Immunology at the Epworth, and the Peter MacCallum Cancer Centre, and Peter Doherty, of course, a Nobel Prize winner. And I remember the first time I met Peter, he was coming in from a cold winter's day and he had his coffee in his hand and his rolled up newspaper, and I just thought, here he is, the everyman with a Nobel Prize. Isn't that what you find in Melbourne? Isn't that a wonderful part of our community? So it's great to be here, and we've got the Roundtable Summit in Canberra later this month, hosted by the United States Embassy and the National Health and Medical Research Council to further improve our collaboration. And I'm going to be part of that, representing the Victorian government. And what happened, by chance I caught Barack Obama's final State of the Union address. And if you remember, he made the challenge to the US. So he said, we want to have a new moonshot, the quest to cure cancer. And he put Joe Biden in charge of mission control. And he didn't say it at the time, but I knew the backstory, that Joe Biden's stated reason for not standing for the Democrat nomination for the US presidency was because his son Beau had died of brain cancer. And so I'm sitting there just watching this, and I thought, we were 6 months off launching the billion-dollar jewel in our medical research crown, the Comprehensive Cancer Centre. And I thought, we should offer to partner America in this. This should be a collaborative thing, not just for one country, but I know how brilliant the minds are and the researchers that we have here. So just treat this off the record, but rather than seeing an idea get lost in a committee, I wrote a newspaper column and got it out there. And the proposition that I put was that it was American ingenuity that put Neil Armstrong, or American exceptionalism put Neil Armstrong on the moon in 1969, but it was Australian ingenuity that brought that iconic vision around the world. And it was through science. It was through the satellite dish at parks outside Canberra. So this was a relationship not forged in war, but in the sea of tranquility. And the measure of any nation's exceptionalism in a globalised 21st century will be the collaborations that we make to save lives. So that's really the theme that we're on about here, and that's what we're looking to do from the Victorian government perspective, to lead nationally and to go internationally and make those collaborations as well. And tonight we're also fortunate to be joined by Peter Doherty, whose research in the immune system played a pivotal role in advancing immunology as we know it today. And we're going to have Jared Roughead here, and Jared to talk about his incredible story, his fight back, and a four-time AFL Premiership player for Hawthorn. And I wish Jared all the best, and I hope he's best on the ground except when he plays Essendon. So this, if you'll allow me that local parochial, very parochial bias, but I think Jared will take that in the spirit that it's meant. He's been a fantastic player, and it's a great story for all of us to learn from. So immunology is considered the next frontier of cancer therapy by helping the body to use its own immune system to fight against cancer. The work you are doing has the potential to save thousands of lives. And the Victorian Government is honoured to be able to support this inaugural conference, and it reiterates our commitment to invest in and to support translational research to find better results for cancer patients right here in Victoria, but to take them internationally. The Victorian Government is committed to improving every Victorian's health status, their healthcare results and the experience. And we're backing that up. So in the 2017-2018 state budget, it included $2 billion in health and ambulance services, programs and agencies over 4 years, delivering better, safer and more services right throughout Victoria. And we're seeing the results through faster ambulance response times, more efficient health services. And it's not just part of what we're doing in this budget, but it's part of a legacy that we built on. And we have a strong history of support. So since 2006, the Victorian Cancer Agency has invested almost $200 million in our cancer research sector, supporting excellence and innovation. And we also have the comprehensive strategy to tackle cancer and cancer-related illnesses, the Victorian Cancer Plan 2016 to 2020. So the plan is to support cancer researchers to collaborate and accelerate the translation of research into clinical results to support a strong and integrated research system. So we've set the ambition of saving 10,000 lives from cancer by 2025. It's ambitious, but I think you have to set ambitious targets. And what we're doing to support this clinical cancer research is we've committed $36 million to the Victorian Comprehensive Cancer Centre Alliance, of which at least $20 million will be to support investigator-initiated and global cancer trials and clinical trials. So the clinical trials obviously are the next critical landmark that we're trying to be able to get the information that we need out of that. And this means more patients getting access to new treatments sooner. And because cancer diagnosis has an impact not only on the individual, but also on friends and families. We know of the ripple effect, and we're trying to address that as well. So intensive and often lengthy treatment and recovery have a whole impact on the workforce, the health system, and the community. So opportunities like tonight allow us to take stock of how far we have come, but also to understand how far we've still got to go and how we need to improve. So we're totally open for your ideas tonight, and we look forward to receiving them. So that's really just the acknowledgements that I want to make, and to wish everybody here to have a fantastic time, to understand that we're listening to what your ideas are. We have some of the most elegant minds internationally involved with this, so you couldn't be in better company. I'm glad to play at least a minor role on behalf of the Victorian Government. I wish you all the best from the Premier, from the Minister for Health, and we look forward to your ideas. We'll connect up with the US and anybody else who wants to achieve this collaborative approach, because that's what enlightenment looks like in the 21st century. Thank you.
So thank you very much, Frank, and thanks to the support from the Victorian Government for medical research in this state. Vital for the health of everyone here. And Frank personally is very passionate about medical research. And one of the things that's always impresses me, Frank, is how you always stay for the scientific sessions when you attend conferences. However, tonight Frank won't, because it's his son's 18th birthday. So he's going to go to his son's 18th birthday. And Frank, we really appreciate you being here and opening this forum on cancer immunotherapy. Thank you very much. Absolutely. Thank you very much, Frank. Yeah. So it's my pleasure to introduce three absolute superstar world leaders in their fields to kick off the evening. So we'll have three talks, then followed by Jared Ruffhead and I will have a chat about his story with cancer immunotherapy, and then we'll open up for some discussion and questions from the floor. But our first speaker is Peter Doherty. Peter has been introduced. I think the thing that excites me most about Peter's work is that the breakthroughs in treating cancer with immunotherapy could not have happened without fundamental discoveries as to how T cells function. And Peter made some of those discoveries. And what is also perhaps not so widely known by many in the audience, but in fact the T cell was discovered here in Melbourne by Jacques Miller. Gus Nossel discovered that one B cell makes a single antibody, which was the forerunner of the monoclonal antibody treatments that we're going to be hearing about from Peter and then also from Professor Sharon Lewin. So without fundamental research in Australia, led by Peter, and also here in Melbourne, we would not have the breakthroughs that we're going to hear about for the rest of the evening. So Peter, thank you so much for joining us tonight and sharing with us the importance of fundamental science in immunotherapy. Thank you.
Thanks. I'm just going to give you a brief rundown on this area, and it'll be a summary, and don't worry if you miss stuff, because I'll probably be garbled anyway, and you'll hear it all again anyway. So basically we're talking about the immunotherapy of cancer, and that is basically using the immune system itself to fight cancer in various ways. Now, the big problem with cancer is targeting. That's the problem with drug delivery in general throughout biology. And traditionally, cancer has been treated with cytotoxic drugs. We poison people, essentially, and irradiation, and that is to kill off rapidly dividing cells. The problem with that is, of course, so the cancer cell is rapidly dividing, so are the cells in your gut, so you lose your hair, all sorts of terrible things happen. It's not very precisely targeted. What the immune system does is it targets immune responses. There's other ways of doing this too. Modern molecular medicine, where we've been able to probe the cells that are expressed in various forms of cancer, has allowed us to develop some very, very specific drugs against proteins, for instance, or mechanisms that are overexpressed in the cancer cell. Particularly would be the tyrosine kinase inhibitors, which were one of the first drugs. These are small molecules, chemicals, drugs. They have various names, like imatinib. Nib, I suppose, means inhibitory. One of the problems for me as I look at this area is the names of the drugs are totally meaningless. And it drives me nuts. And it often takes me a long while to work out what the hell they are, because you have to look them up. But that's the way the world is. So what we're going to use here, though, is the exquisite specificity of the immune response. Now, of course, Melbourne is a great centre for immunology research. And the father of immunology research in Melbourne was Sir Macfarlane Burnet. And so we mentioned Gus Nossel, Jacques Miller, me. We're all, to some extent, grandchildren and children of Macfarlane Burnet. And it really speaks. Australia has been a leader in immunology over many, many years. Now, my particular research has been concerns immunity to viruses. We know that there are viruses can come out of anywhere, they challenge us, they infect us, and we don't have pre-existing immunity. So we need a very broad immune system to deal with viruses and bacteria and so forth. Totally unexpected. And the immune response we're talking about is what we call adaptive immunity. That's the part of the immune response we stimulated with vaccination. As you know, that's very specific. If you're vaccinated against polio, you're not protected against measles. If you have measles, you're not protected against mumps. We all realise that immunity is very, very specific. And of course, we've got something very specific, means it's very precisely targeted. So that's what we're talking about here. And the immune system, we think, has evolved to deal with infection. The word immune itself comes from the term immunus, which means without tax. The way President Trump's America wants to work. No taxes on the rich. And the tax we're concerned with here is the tax of infection, the tax imposed by very simple organisms which multiply very quickly and change very quickly, whereas we neither multiply very quickly nor change very quickly. So we need this very diverse and complex immune system to deal with them. And those immune cells, or immunity, all works, or adaptive immunity, all works through the white blood cells. Various types and categories of white blood cells, we call them T cells and B cells and various other cells as well. There's even a cell called NKT cell, which is really quite strange. But Dale or David or Arnie or whatever his name is works on them. I won't say any more about them. We have a running joke about this and their relevance. They're tremendously important, of course. So all adaptive immune responses start because we've got cells with specific receptors on their surface. They may be the immunoglobulin molecules that become the secreted antibodies that float around in our blood and will grab hold of an invading virus. Or they may be the surface receptor on the T cells that I study and Jacques Miller discovered many years back. So there are very few cells with an individual specificity. So what happens in an immune response is in the lymph nodes, these glands that swell up in your neck, for instance, they multiply. They what we call clonally expand. They're clones. They're clones of cells. They keep multiplying and multiplying and multiplying. Some of them become the effector cells that will secrete antibodies or will kill off other cells or do the various jobs. And some of them become memory cells. And memory means they stick around. And some of these memory cells in both the B cell and the T cell lineages can stick around for 50 years or more. They're truly extraordinary. And a lot of our research over the years has been with trying to understand that memory better, and there's an enormous amount going on as well now. And if you've got memory cells, you both have more cells that can respond to something that comes along, if the same thing or a cross-reactive thing comes along again, and also you can expand them a lot quicker to become the effector cells that do the job of immunity. So we're interested in immune memory and immune effector function. Now, all immunity, or the immunity we're interested in here, mainly works by protein-protein interactions. That's the proteins that are the antibody molecules or the T cell receptors, and the proteins that are expressed on viruses or bacteria or bacterial products and so forth, like toxins. Some of the antibacterial vaccines are antitoxins. And they, particularly the antibodies, which are secreted, float around in blood and stay there for years, made by cells like plasma cells and so forth. These are factory cells. If you don't understand any of this, you'll get better explanations from other people who'll be much more coherent and much more attractive. But many of these new new drugs, we call them, I prefer to call them biologicals because they're not synthetic chemicals, are actually antibodies. They're the specific antibody made by an individual clone of cell, and we can grow those out and we can immortalize them in various ways or produce them synthetically by various ways, and they're essentially monoclonal antibodies. So you will hear in this about various drugs, though I think biological is a better term, where the last three letters are MAB. So anything that has MAB in the last three letters, it's not MAB Queen of the Fairies. It's actually a monoclonal antibody. And they have ridiculous names like Plumabab and Fosubabab, and I can make them up. But if it's a MAB, it's due down to these two guys, so Cesar Milstein and George Kohler, who developed this technology way back. They won a Nobel Prize for it. They're both gone now, but that's really a fantastic thing. And some of these monoclonal antibodies are targeted against molecules that are overexpressed on various sorts of cancers. Now, these may be what we call oncofetal antigens, for instance, something that's expressed in neonatal life, but is not normally expressed in adult life, but emerges again on a cancer cell. And some of those things are direct molecules that have been used to target a specific cancer. Others of these molecules do things that are rather more indirect and rather marvellous, and you'll hear more about that. Here are some of these things. A lot of them are approved as therapeutics on our PBS. They're very expensive products because they're proteins. They have to be grown up under what we call GMP conditions, good manufacturing practice. And also the drug companies, you might not suspect this, but they make a hell of a lot of money and have a hell of a big markup on it. We all know drug companies are really nice, but you know, they need a lot of money. I mean, I visited a drug company once and we were late for lunch and we had to take the helicopter, and that's not cheap. But hundreds of these things are being produced all the time. This list here, there's an enormous amount of effort going on, and there are some really, really very promising products, which is one of the reasons we're really getting cancer more and more on the back foot. One of the big problems for it is the cost to healthcare systems. That's a really big issue and a really big difficulty. And this is a list just of monoclonals that were approved by the American Food and Drug Administration, the primary registering agency. We hope it survives President Trump. And that's just between 2015 and 2017. So you can see how many of these things are coming onto the market. Now, just say a bit about vaccines. In my area, of course, the great triumph of immunity was always refers to the vaccines. We have a lot of vaccines that are very good. We don't have vaccines against some terrible diseases. You'll hear from Sharon Lewin, who works a lot on HIV/AIDS. It's been a very, very difficult problem. But we do have some cancer vaccines. And one is against the virus called hepatitis B virus. Now, hepatitis B virus doesn't directly cause cancer itself, but it sets up in the liver a chronic disease with chronic inflammation. And chronic inflammation is really, really bad in all sorts of circumstances. And we're realizing more and more that many degenerative diseases are associated with chronic inflammation. So anti-inflammatory drugs are also becoming more and more important. And some of those are anti-immune reagents. Like TNF in rheumatoid arthritis. What happens is the inflammation in the liver causes so many liver cells to multiply as a sort of a bystander that many of those will transform and become cancer. So this has been an extraordinarily successful cancer vaccine. Many people have worked on it. And then, of course, we're all aware of the human papillomavirus vaccine. This is a virus that actually directly causes cancer. And that vaccine, developed by Ian Fraser in Queensland with various colleagues, is now, of course, this wonderful anti-cervical cancer vaccine that's used in humans. There are some other anti-cancer vaccines in animals, but we haven't translated those technologies across into humans as yet. The question is, can we make vaccines against tumours themselves? Now, I'm going to talk about the killer T cells. These are the cells I work on. This cell is going to kill that cell. This is a video taken by Misty. Misty is going to speak at this, and you can see it's killed. Now, I won't show it again because Misty will show it, and I think Sharon might show it, but it's a really good video, and I love it. I sit and watch it at night at times. My God, it's better than the ABC at the moment. And these are the cells that I've worked on for 50 years. And Misty is actually one of our students and a wonderful, wonderful scientist. She's now at the Walter and Eliza Hall Institute, where she has her own lab. And these are the killer cells. They're the hitmen of the immune system. They're the cells that go around the body looking for cells that are abnormal and need to be knocked off. Now, they do normally keep some forms of cancer under control. We think they keep melanoma under control for a time. We think they also can keep some forms of skin cancer under control, renal cell carcinoma. But the cancers often can mutate away from that. And that's a problem. And then you get the problem with the tumour. This is the type of immune response you see. You get that clonal expansion of cells when they're stimulated. And if you could do that with a vaccine, maybe you could generate effector cells and memory cells that would stick around. And then when you re-stimulate, it comes back again and then maybe knocks off your infection or your tumour or whatever you're trying to deal with. And also these cells sit around in the tissues. We call them tissue-resident memory cells. A lot of research going on in this at the moment with some very good people, particularly at University of Melbourne. This is Scott Mueller. One of our graduate students went to the States, worked with some top people, came back, set up his own lab. And these are what we call resting cells. If you saw the little video part, I'll play it again, you can see that they're not really resting, they're sort of jigging around all the time. These are the cells that can potentially be expanded again. And what really intrigued us for many years was we knew a lot of these cells were sitting around in tumours, but they weren't doing anything. And people tried to take them out and they tried to re-stimulate them and inject them back in, all sorts of things. They got very, very limited success. And really, the great triumph has been the discovery that you can wake those cells up again. They're specifically turned off And by using some of the monoclonal antibodies, you can target the molecule that's involved with turning them off, and you can turn them back on again. And this has been an enormous triumph. This is what, for instance, cured Jimmy Carter of cancer. Well, you know, nobody's ever really cured of cancer. The only way you can be sure you're cured of cancer is when you die from something else, right, Grant? Yeah. So that's what my cancer friends used to tell me anyway. So, but they've been enormously, Ron Walker, for instance, got many more years of life due to the fact that these drugs were used to get rid of his melanoma. As we know, he eventually succumbed. But these immune checkpoint inhibitor drugs are really, really enormously important and powerful. But they're only curing about 20% of cancer. Now, you might think that doesn't sound like much, but if you're a cancer doc and you have 20% of your patients who you knew absolutely would die, and they don't die, and they have a much longer life, then that's a fantastic advance. Now, so the big thrust at the moment is to try and get that 20% level way up so that we get a much higher success level with these. I also, the guy who gets the real credit for pushing this along is Jim Allison. Jim first gave his presentation on this, a basic science presentation, at an international meeting in 1995. And so that's the sort of time frame you're talking about often, to get something from discovery and initial work to actually clinical application. You're talking about 15 or 20 years, very commonly. So you've got to be prepared to fund research, fund development and application and clinical trials through that period to get it to market. So it's a big ask, and it takes a lot of money. The other technology that you may hear something about is what we call the CAR T cell. This is where we take these killer cells, but we engineer a receptor made from the antibody molecules into them. That gives them very specific targeting too. Unfortunately, we don't fully understand how these work. They're very promising. They can be used to control some forms of disseminated cancer, but they're not good at going into the cancer and knocking off the cancer cells themselves. So there's a way to go there too. So lots of research to be done, lots of challenges. Here's just a few questions. Can we really make cancer vaccines? If we could make cancer vaccines, could we combine that with the removing the blockade and turning them back on and making them really work well? Can we get these CAR T cells to invade solid tumours? Lots of research that can be done, lots of things happening, but it's a very exciting time for cancer and cancer research, and we're really getting on top of a lot of diseases which were formerly really enormously intractable. So thank you very much.
Thanks, Peter. Just an absolute brilliant summary of the advances in immunotherapy. One of Australia's great iconic scientists, Peter, thank you so much for sharing that with us. Next speaker will be Professor Sharon Lewin. Sharon is the director at the Peter Doherty Institute for Infection and Immunity and an absolute world leader in the field of human immunodeficiency virus, HIV, the virus that causes AIDS. Because these immune therapies, and Peter started to describe how they work, Sharon will expand on it, can be used for more than just cancer. Maybe we can cure HIV as well, Sharon. Over to Sharon.
Thank you, Grant, and fabulous to be here and to see so many people interested in immunology. As Grant mentioned, I direct the Peter Doherty Institute, named after the one and only Peter Doherty. So always a very hard act to follow when you have the doyen tell us about immunotherapy. But I have the great privilege then of singing some of Peter's praises, not that he put me up to this. But first of all, the Institute only just opened in 2014, has over 700 scientists just working on infection and immunity. And so for people in the audience, they might be thinking, well, what's that got to do with cancer? Well, you just heard a whole lot of new cancer therapies that came out of our understanding of how the immune system works. And for years, we've been looking at this, especially in relation to infection. So I'm going to talk in a little bit more detail about how specific type of immunotherapy work, something called immune checkpoint blockade. Talk a little bit about where it sits in cancer treatments. I'm not a cancer doctor, I'm an infectious diseases doctor, and I've spent most of my career working on HIV. And I want to end with talking about how these new developments are really relevant to viruses that we can't cure, like HIV and hepatitis B. So, as Peter already told you, the immune system is designed to recognise things that are foreign. I'm going to show a slightly different video, but it's also Misty's video, and it just, if I can coordinate the arrow. I can do all these other things that I can't turn on a video. I can't turn it on, but what you can, what you might want to see, let me just have another go at this. Why can't I? There we go. So Peter's already shown this to you, that we've got this green cell, which is a killer T cell, and it can kill tumour cells, and this is what you see is turning red. What Peter didn't tell you about is this is not a random event. This happens under exquisite control so that your T cell recognises something foreign with exquisite specificity. And actually that was the work that Peter won the Nobel Prize for in the mid-70s. So when he was talking about this long progression to go from a discovery to a treatment, we're actually talking about the mid-70s when Peter's work told us that these killer T cells recognise something foreign with exquisite specificity, through to the mid-90s when we began to get drugs that woke up these killer T cells, to now where we have treatment. So the timeline of scientific discovery to cures is extremely long, and we need to keep investing in it. So exquisite specificity. But we want to make sure that these killer T cells are also very carefully regulated. And this slide comes from my colleague Dale Godfrey, who's done a lot of fantastic work in this area as well. And I think this is a beautiful illustration of what happens. So these killer T cells have to be regulated. And normally the immune system is very carefully controlled, so these killer T cells don't go around knocking off normal tissue. In other words, it's like the handbrake on those cells is turned on. If the handbrake is released, though, that means you could get that immune system fighting a bit harder to recognise something like cancer. And that's exactly what the discoveries are that we're talking about, how to temporarily release the handbrake on the immune system, leading to new ways to treat cancer and maybe to cure chronic infections like HIV, which is the area that I work on. So just a little bit more detail. When someone has got cancer or when you're infected with a virus, bits of the cancer and virus are presented on the surface of that cell. It's a sort of way of saying something is foreign in the body. And along comes something called a T cell, which is specifically designed just to recognise that unique blue diamond shape. Every single T cell has a different a molecule that recognises something foreign. So you can imagine how many different types of T cells we must have to recognise all the different foreign things we contact. Once there's that recognition, the T cell's basically turned on. A few other molecules need to bind to really rev up that T cell and put it into action, and it becomes a killer T cell and kills its target. But we need ways, the immune system also has ways that it has to turn that T cell system off, because you can't have this, this killer T cell running around killing other things. So these, what we call immune checkpoints, they sort of put the T cell into check, get turned on, and this basically turns off the immune system and the handbrake goes on. So when we talk about immune checkpoint blockade or these magic new, incredible new drugs that have transformed the outlook of cancer, or immune checkpoint blockers, these are molecules that bind to these immune checkpoint markers. The two that was first developed, as Peter mentioned, was anti-CTLA-4 ipilimumab, that was the first one, and the next one was anti-PD-1 or nivolumab. And what these antibodies do is they block those immune checkpoint markers, release the brake, and therefore you can turn back on that T cell for it to become a killer T cell. And that's what we mean by checkpoint blockade immunotherapy. And this is really the big advance that has transformed what was previously universally lethal cancers. So you'll hear more from people that treat cancer, but basically this is the main shift that we're now seeing. Is historically we used to always design treatments that target the tumour cell, so surgery to cut it out, radiotherapy to poison it, chemotherapy to eliminate it. And now we're targeting treatments to the immune cell and then taking advantage of our own immune system to do the job, which is, if we can get this right, because we are interfering with a very tightly tuned system, this is a far more sort of natural way of eliminating something foreign. And this has led to the development of a whole new line of drugs, as we heard, anti-CTLA-4, anti-PD-1, anti-PD-L1, all got these ridiculous names, as you learned about from Peter, ending in MAB because they're all this type of antibody. And this is just one example of what these antibodies can do. This is the patients who survived with metastatic melanoma who received one of these drugs, ipilimumab. That was the first kid on the block. Survival of 34%, which was remarkable because previously this was a universally fatal disease. Nivolumab survival 50% at 3 years. And then now we're learning if you use these drugs together, you can even get even better survival. Unfortunately, you get worse toxicity, but you get better survival. And there's a whole lot of other different cancers that now are responding to these immunotherapies, which is what all of this excitement is all about. Now, very quickly, how is this relevant to HIV or hepatitis B? So for both of these chronic virus infections, we have excellent antiviral treatments, but we don't have a cure. So what happens with HIV or hepatitis B? When you're infected, you have lots of virus in your body causing lots of damage to your immune system if it's HIV, to your liver if it's hepatitis B. You put people on treatment and the virus disappears from blood, but when you stop the treatment for both HIV or hepatitis B, the virus rapidly comes back. In HIV, it happens in about 2 to 3 weeks. So my interest over a long period of time is understanding where that virus hides and how to get rid of it. And we know there are 2 forms of the virus, an actively replicating form, and we can easily get rid of that form of the virus by antiviral drugs. But there's also these other forms of the virus, which is actually very similar to cancer cells, where the virus gets inside the cell and basically goes to sleep, so is no longer recognised by the immune system or the antiviral drugs. And what we're doing with respect to HIV infection is a very similar sort of model to what's going on in immunotherapy. We do have antiviral therapy that can eliminate these productively infected cells, but we're now using immunotherapy, trying to boost the immune system to recognise and kill latently infected cells. It's not all rosy though, because we're fiddling with this very tightly tuned system. The current drugs don't get it right completely, meaning that there are what we call autoimmune toxicities from immunotherapy, where these drugs also dysregulate your immune system and it does start reacting against itself. So you get a whole range of toxicities, skin rashes, hepatitis, gastrointestinal toxicities. And we're getting better, much, much better at managing these. The initial drugs had far worse toxicities and the newer drugs are going to get better and better. This is an issue for when we're trying to use these drugs in the setting of HIV, where people have actually got a normal life expectancy, very different to cancer. But there's a lot going to happen in the future. This is an example of all the immune checkpoint markers we knew about back in 2011, and there's many more to add. CTLA-4 and PD-1 are really just the first in which we've got antibodies to, and there's antibodies in development for many other of these immune checkpoint antibodies. So we'll see a bigger range, a whole suite of antibodies to choose from in the future. And finally, as Peter said, immunotherapy is not just about immune checkpoint blockade. I want to talk about that in detail because that is really what's revolutionised things. There's other strategies. At the moment, those drugs that I mentioned are all, you have to give as injections, but we'll get drugs that are oral. We can deliver immunotherapy through viruses, CAR T cells, Peter mentioned, various forms of antibodies and cytokines and vaccines. And some of these are already registered and approved for use in the clinic. So just finally, scientists have uncovered that the immune system is critical in controlling some, but not all cancers. The temporary release of the natural brakes of the immune system is highly effective in eliminating some cancers. This might work for chronic viruses like HIV or hepatitis B, not yet proven. And if I can ask you to remember one thing from tonight, discovery research from over 30 years ago was a critical part of how we have effective treatment for some cancers today. So we really need to keep funding discovery research to have effective and safe cures for all cancers in the future. Thanks very much.
Thanks, Sharon, and fantastic presentation, so clear on what has been such a major breakthrough in cancer and the hope also for controlling infectious diseases. And aren't we pleased that Sharon's leading an institute with 700 scientists to continue those vital discoveries. So our next superstar speaker, before we move into the second part of the session, is Dr. Cassian Yee. Cassian is at the MD Anderson Cancer Centre in Houston in Texas, the world's largest cancer centre, where he heads up the cell therapy program. And we've heard about these immune checkpoint therapies. That's the breakthrough now. But 2017 was a really special year for cancer because the FDA, you heard about that from Peter, Food and Drug Administration, approved for the very first time a cell therapy for cancer. And Cassian Yee is one of the pioneers in this field, and we're really happy to have him to tell us about this exciting technology tonight. Cassian.
Thank you, Grant, for a very kind introduction. I just want to say that it's a pleasure to be here in Australia. Even though I live in the US, I'm Canadian, and in light of the current administration, I'm holding on to that passport. Actually, I do know how to prepare for this. Give a very broad overview. It's not going to be specifically about cell therapy, but we'll include overall some concepts in how we might fight cancer using the tools we have now available, immune checkpoint therapy, as you've heard already, and adoptive T-cell therapy, which is something I've been working on for about 25 years, and just at a time when you could fit everyone who was interested in immunotherapy in a bathroom. Now it's completely overcrowded and standing room only, so it's been a real fortunate confluence of circumstances. And so I'm going to introduce this as a green eggs and ham sort of analogy in which there are, in fact, 3 pillars of cancer therapy. You've heard of already surgery for early stage cancer, which can be curable, radiation therapy, and then certain types of chemotherapy and targeted therapies, like the imatinib you've heard mentioned from Dr. Doherty. Really there are 4 pillars, the last being T-cell therapy or immunotherapy, and I guess broadly speaking, this is the green eggs and ham of clinical oncologists, meaning, you know, it's something we like to try, but maybe not really. And I think that basically it's come across not only for CAR T-cell therapy, immune checkpoint inhibitors, and something that my lab has been working on, which is growing naturally occurring T-cells. I'm not going to get into a scientific discussion so much. This is to sort of point out the fact, I have one too, actually, so everyone's got one. I lifted this from the internet, so you can get this anywhere. But basically, a T cell chews up the tumor cell and blah, blah, blah. And you've heard, if I can get to the next slide. Oh, yeah. And this occurs through the T cell receptor, remarkable specificity. Again, work that was pioneered by Dr. Doherty and Mike Bevan and others, showing that this recognizes the peptide in the context of the MHC molecule, and the target antigen is brought up to the surface so the T cell can see that and then release toxic granules, punch holes, and literally destroy the tumor from within. This breakthrough really in recent years, as you've heard mentioned, was both combination of adoptive cellular therapy and also immune checkpoint blockade. It is a paradigm that combination therapy is important. But to touch briefly on immune checkpoint therapy, which everyone has heard really started out in melanoma, has begun to spread to other cancers. And this is really quite remarkable how a single drug can be used to treat multiple cancers. And as a clinical oncologist, I can tell you this was not the paradigm. You would use certain combinations for a particular cancer, but as you know, we're treating the immune system, we're not treating the tumor cell. However, it should be kept in mind that adoptive cell therapy, I gave away the punchline there, is important for treating patients. And this drug, which I can't pronounce either, is a type of CAR T-cell therapy, an engineered T-cell that recognizes B-cell leukemias and B-cell lymphomas, but can be very expensive. And unfortunately, that may be prohibitive in terms of making it broadly available, and perhaps it's a point of discussion we can have later. They did a study in terms of years of life gained. This is actually a bargain, but it's hard to imagine that being reasonable, considering that, or reasonably available, I guess I shouldn't say that, in terms of reasonable. It really is something that should be considered when we're bringing this out to treat more patients. And I think what we do need to do is look at this as a integrative type of therapy involving not only immunotherapy, but also as this little tumor here surrounded, actually, they've co-opted the immune system to protect itself. It's got a stroma around it that contains regulatory cells, suppressed T cells, and so when you grow the T cell by adoptive cellular therapy and transfer that and expand it and then maybe energize it with drugs and immune checkpoint modulators, only some of them can actually get into the tumor cell itself, and that invasiveness of the T cell into the tumor, infiltration of the T cell in the tumor is very important. And this actually in recent years has shown that may be important to treat not only the tumor with radiation and drugs, but also certain molecules. They call it kinase inhibitors, for example, which can soften up the tumor and allow these T cells to migrate into the tumor and take care of a problem for the long term. So I think this sort of approach is both exciting and friendly. I think Greg Hunt mentioned this this morning, and I'm going to paraphrase that a little bit here by saying some of the important points for addressing in terms of moving immunotherapy forward as a treatment modality, not as a boutique therapy for specialized groups of patients, but as a treatment modality. Really, one is expense, obviously, but not just the expense, of course, of growing the drugs and making these T cells and making antibodies, but of administering them as well. And along with that are the toxicities, which are brand new to many of us who've never seen patients receiving immune checkpoint therapy before. These autoimmune side effects, which may just be rashes and colitis. You saw the whole spectrum of these possible immune toxicities. We've got the endocrinologist involved, the rheumatologist, everybody helping us to sort of give us signs of what might be potential toxicities so we can treat them adequately and provide the most effective and manageable care of patients, not only in the tertiary care setting like MD Anderson Cancer Center or other major centers, but also in the community. The availability is important, and then how does this change the standard of care? Is this a new paradigm? It certainly is, and when is it applied? How do you apply it in the context of all the available care that's available, and then providing this out to the community as well. And certainly one thing I'll emphasize again is the timeliness and critical need for clinical trials as any new therapy comes forward, funding for such trials, and then harmonizing of these strategies and priorities with government and regulatory agencies to ensure that it's a safe and effective therapy. And hopefully, that was the last slide. That was a good one. There we go. I hope. Oh, well. Somebody's going to scroll this for me. There we go. And so thank you very much for your attention.
Thanks very much, Cassian. One of the real stars in the world of cancer immunotherapy around the globe. So now we'll move into the second part of the evening, and I'd like to welcome Jared Roughhead up to the stage. So come and join us, and Jared and I will sit down. And Jared and I will have a chat about his. Good to see you, mate. Jared and I will have a bit of a chat about his experience with cancer immunotherapy, and then we'll open up to our panel, and you'll be able to ask some questions of our panel, and we've got some extra experts as well to introduce. But Jared, fantastic that you're able to join us here tonight. So you've been hearing a lot about all these cancer immunotherapies, but you know, at the heart of everything is of course the cancer patient. And you know, you were travelling pretty well, right, mate? You had 4 flags with the Hawks, yep. And life was pretty fantastic. So what was it? How did you feel when that diagnosis of melanoma first came along?
The first one with my lip, I didn't think much of it, to be honest. Being a boy from the country, red hair, fair skin, you obviously know that you're not great with the sun, so I was pretty diligent in terms of covering up, sunscreen, hat, everything like that. But I had what I thought was a blister on my lip, and it probably lasted a little bit too long. I finally got a punch biopsy done on a Monday afternoon, and Wednesday morning I was getting it chopped out. So it happened pretty quick, as they say, with the ones on your lip. They grow like a carrot compared to ones on your back that spread. So it's weird, but with footy you treat it like an injury, so they said you could be out for 1 or 2 weeks, and that's how I dealt with it. I was back playing within 2 weeks and everything felt fine. So when we got to the one in 2016, I just got married in January.
And we were watching you with scans, right?
Yeah.
One of the reasons for that that you were having the regular scans is that, interestingly, the science tells us that the immune system works better if the cancer lumps are small. So if it was ever going to come back, we wanted to catch it early. So then what happened in 2016? Yeah.
So I'd had a couple of PET scans after the initial lip one, and they'd come up clear. We got to the one in, I think it was the second or third PET scan post my lip, and as I said, I'd got married in January. Everything was fine. I had another injury that I was close to coming back to playing, and then I was having PET scans on a Monday. The following Monday I'd meet Grant for a report, and I walked in and he said, how's your knee? I said, yeah, fine, I'm hoping to play next week. And he goes, oh, we're in a bit of trouble here. And initially I thought, I've just got another spot on my back or another freckle that probably needs a bit of checking. But when he said, no, you've got 4 spots in your lungs, I didn't know really what to think. Your mind goes into shock a little bit, and you know, first thought was, this may seem weird, but how long am I going to miss a footy? Ever since I moved up at 17, that's all I've known. It's like, right, play footy, do school, play footy, and when he said, oh no, you could be out for the next 2 years, you know, I really didn't know what to think. So from there, I went out to the footy club, tried to tell. I couldn't ring Sarah straight away, so I rang one of my teammates. We came out and tried to work as a team what we were going to do. The next day, we all met together and worked out the best treatment from there.
Yeah. And what were your first thoughts? Did you think, I'll need chemo, or have you heard about cancer treatments, and did you have any preconceptions about what treatment you might get?
I suppose going into it, I'd only heard of chemo, radiation. There was talk of radiation with my lip initially, but that didn't need to happen. So then, when you said we're going to try something new, I was like, all right, let's go for it. I'm a blank canvas in terms of all this. Let's attack it, and that's how we went about it. I'd had a biopsy on the lung where they took one tumour out I'm good with dates. So Monday the 23rd of May they took it out. A week later I was having my first infusion of the drugs.
Yep. And had you ever heard of immune therapy? Before I had to start to talk to you about immune therapy as the way to go.
My wife works at Swiss, so I knew of immune tablets. That was about that was about it. But no, not until you said this is something new. This is something that is positive, and we think it'd be really good for you to trial.
Yeah, and there was. And I won't test you to see if you can repeat the names of the drugs, can you? Or
Yervoy and Opdivo.
Ah, yes, yes. Easy to say than the ipilimumab and nivolumab.
Yeah.
So you had the two treatments, which Sharon showed us in her slides how two treatments gives better survival than one. And at the time, we didn't know how much better they were. We knew they gave more side effects. There were some early signs that they were better. And so did you have any fears really about whether this was the right way to go?
No, I wasn't too concerned at all. That's why I trust you. I'm not an expert at this. If it was about footy and you asked me how to kick it, I reckon you'd come to the right bloke. But
I tell you what, mate, you'd need a lot of work with me.
But when it came to this, it was like, I'll listen to whatever you say. Obviously, Dr. Google I never looked at. Sarah made the stupid decision to look at that a couple of times, which is bad. But yeah, I was right. Let's go for this and let's attack it as hard as we can, and that's how we viewed it.
And look, I think, Peter's made a really important point about the science of the immune system. So, the T cells can hang around for 50 years and have memory and keep attacking the cancer. So did you anticipate that you'd get a really good result? Or what was what were your thoughts?
Obviously, you go in asking questions, because when you hear the word cancer and melanoma and with what had happened with Jim Steins and stuff like this in the past, if this is, when did it happen? 2016. So if this was 5 years prior, I may not be here. Mm-hmm. So when you put it like that, I've got nothing to really lose in that sense. I'm 29. I was 29 years old at the time, and it was like, righto, as you said, I hope to see you to live to be a granddad, and that's what that's how I viewed it. So that's how we went about it.
Yeah, fantastic. And the theme of tonight is turning fear into hope, power of anti-cancer immunotherapy to restore a healthy lifestyle. So we will get to healthy lifestyle as an elite sportsman at the end, and you've certainly achieved that, which is fantastic. But just wanted to ask you a little bit about the treatment. So, so you actually only had 3 treatments.
Yep.
Yeah. And why did we have to stop, and what did you go through?
So the first one, I was having them 3 weeks apart. The first one, I had no worries. Obviously they do it over a long period of time, so you don't get any side effects at that stage or anything. And then I had a 3-week window where I felt pretty good. Grant said you can go away, so my brother and I went to the NBA finals in 16 just to get away. Came back, had the second dose, and the next day I was gone. I felt like I had a really bad flu. I was sweating, and it probably took, I don't know, 3 or 4 days to maybe get over. I've never been one to stay in bed with a flu or anything like that, but I was bedridden for 3 days. Probably took the full 3 weeks to eventually get over everything. We'd had a couple of blood tests in between, and Grant said, "You're probably best off to go dry for a little bit," which was interesting. First time probably since I was 17.
Yeah, because there were some slight changes in your liver tests after 2 treatments, weren't there? So we have to be careful with alcohol. Yep.
And a couple of other things. My eyes started to go a little bit red. I wasn't on the other stuff. It's just a side effect. And my lungs were a little bit iffy too. So there was three things going on there, and they weren't major. We had the third dose, and I thought that I'd be okay, and I was for the first couple of days, but then, again, good with dates. So we played Richmond. It was Sam Mitchell's 300th, and I don't remember being at the game. I'd walk. It felt weird, but it was cold and wet the day before, and they're seeing me walk out on the oval, just walking in the wind and the rain, and they're like, "What are you doing?" And I said, "This is, it may seem weird, but this is the only thing that's making me feel alive, is having the wind in my face and just something to make you feel like you're still here." And then the following week we travelled to Perth, and I wasn't too bad, but my feet started to go numb, and I couldn't sleep. I woke up in the middle of the night when we were in Perth, and I had to go run the cold bath and stick my feet in every half hour to an hour to just try and... It was like I had weird pins and needles or weird frostbite that just seemed to be eating away at my feet. And I kind of said to Grant, if I'm going to get back to playing footy, I kind of need these. I need them to be feeling all right. And so at that stage, when there was 4 different side effects, we kind of said... you're pretty full in terms of the drugs, I think.
Yeah. So Jared, you had inflammation of the lungs.
Yep.
You had inflammation of the eyes, inflammation of the liver. That's why you had to go dry.
Yep.
And then, the one that we were most worried about was actually the feet, because the nerves were inflamed.
Yep.
And so we gave you a lot of treatment to try to, turn that around. Do you remember? Do you remember the treatments that you had?
Yep.
For the nerves. Yeah.
So one was immunoglobulin.
Yep.
Which is clear blood.
Yep.
Doing all right here.
Honorary medical degree at the end of the session, I think. Yep.
I'll wait because I listened. So the second one, we had a plasma exchange, which would take a long time. Being a little bit bigger, you'd sit there and you'd go through 2 big, I think 4-litre bags. And they initially worked, but I'd still have to sit there at the plasma exchange and have my feet in a bucket of water to make them feel okay. And then you tried one of the mAbs, the rituximab, I had 2 doses of that, and it stopped so what had happened with my toes, it had eat probably back to mid-foot, and it stopped there after I had the rituximab. So that was... I think 5 weeks after we had the last dose. Yeah,
and so we gave you lots of treatment for the nerves because, as you say, a footy player, you sort of need your feet, don't you? And you've got to be able to kick. So how are you now with the feet?
Yeah, so initially I went and saw Mark Farragher, who's, what is he?
Neurologist.
And he did a couple of nerve conduction tests on me, and just made sure... or, confirmed what was going on. And so after the rituximab, I felt pretty good. I started pre-season at the end of 2016 and felt all right. Thought I was probably 90-95%, but even towards, or probably at the midpoint of the season, I'd got back and played every game up until... I played every game last year, but until the mid-season break... even some coaches said, you're not kicking the ball as well and you haven't got the agility of what you used to have. And probably until the last maybe 5 or 6 games, and even now I've probably got, with this pre-season, a lot of coaches have said, gee, you're looking a lot better than this time last year and you're moving well. So without even knowing, I'd say I was 90-95% to them, but maybe I was only 80-85. So you did tell me that a nerve grows back 1 millimetre a day and starts here. So... I'm looking at a fair few days.
You're not the shortest bloke going around.
I've grown back. Now I'd say I've got full feeling and no issues. I used to have trouble walking on bitumen and different types of surfaces, whereas now I'm fine.
Yeah, so it's pretty interesting. So return to normal lifestyle. Basically, all those 4 side effects with treatment have now completely recovered, right?
Yep.
And we'll get to other things happening in your life, but footy season's about to start, and you're right to go for the Hawks fans in the room.
I was. We had the captain's day today, which is always a great day. but yeah, ready to go. I've done all of the pre-season. I haven't had any issues since you gave me the all clear.
Yeah, that's great. So just one more question before we'll talk about, you know, where you've got to. So you've had what we call a complete response. So, you know, nothing on the scans. How do you feel when that, you know, you got that result, that the complete response?
Mate, it's better than any grand final I've won.
Wow.
Because as much as footy's 5-10% of my life, this drug's given me 100%. So, you know, I remember you sat down and you said, right, you've had a complete response. I expect you to live to be a granddad now.
That's right, and it gets to the, you know, Peter's point about T cells lasting for 50 years. So they've got memory and they keep controlling the tumours. So there's a very, very good prognosis. So, you know, getting back to healthy lifestyle, some great things have happened to you. So captain of the Hawks now, which is fantastic. And a very recent event, 4 or 5 weeks back. So tell everyone about that.
I've got a chance to be granddad. So I had a daughter 4 weeks ago.
Yeah.
Little Pippa came along. She's been amazing. I don't worry about the little things anymore, even though I probably said that I didn't when I had cancer. This definitely takes your mind off things, because even if I've had a crap day at the footy club, she definitely doesn't care. So the last 4 weeks have been unreal, and Sarah's a superstar as well. That's my wife. She's done an amazing job as well.
Yeah, so that's fantastic. I mean, you know, one of the amazing things with the immune system is we're able to turn it on, eradicate the tumours. They all went away. Yes, they had side effects, but they've got better, and now really, back to 100% in every respect, would you say?
Yep.
Yeah,
yep.
Fantastic. And that's, you know, that is, you know, not what tends to happen with cancer treatments in the past. So, you know, we had a survivorship conference recently at the joint conference with the Victorian Comprehensive Cancer Centre and the Peter MacCallum Cancer Centre, and only 20% of people after cancer treatments get back to 100%. So that's what's different about these immune treatments, you know, harnessing the body's own immune defence against the cancer in a way that, you know, can have a lot less side effects. But of course, you know, as Jared showed us so clearly, you know, the side effects can come up in the short term, but they are treatable if you with diligence. So I think it just remains to thank Jared for sharing a remarkable story. Well done, mate. Stay. All right, so we'll move on now to our panel discussion. If I could have the panellists come up and join us. Some of them you've already met this evening. Alan and Cassian and Sharon. So you've met some of our panel, but we've got another couple of panellists as well to introduce. And the gentleman in the red tie is Professor Miles Prince. So Miles is one of the pioneers in Victoria of cell-based therapies and leads the precision medicine immunology program at the Epworth Hospital and is also at the Peter Mac. Welcome, Miles.
Thanks, Graham.
And on Miles's left is Dr. Rebecca Del Conte. I can call you Doctor now, Rebecca. You graduated?
In a couple of weeks.
2 weeks, okay. So Rebecca is therefore still formally, I guess, a PhD student, even though she's passed. One of the real rising young stars in cancer immunology in Melbourne's. And her area of expertise we haven't heard about tonight, natural killer cells, and we'll ask her a little bit about natural killer cells a little later, but sounds, you know, a bit intimidating, but a really interesting other component of the way the body can fight cancer. So thanks very much for joining us, Rebecca. All right. So we'll open up for your questions, and so please, we've got some microphones. And so if you just put your hand up and come to the microphones if you've got a question. But to sort of kick things off, I might ask our panellists some questions. And why don't we start with Rebecca? So, Rebecca, after some patients respond to immune treatments, some patients get a complete response like Jared has, and the prognosis for Jared's fantastic. But sometimes the tumours don't go away completely, they shrink. And in a fairly small subset of patients, they can start to grow again. So your research, natural killer cells, could that be the key to helping that rare situation or that uncommon situation where tumours start to grow again after immune treatments?
Yeah, so I think natural killer cells are definitely an area of immunology that is rapidly generating a lot of interest. And as you've heard a lot tonight, I mean, fundamental biological research is really, really important for these type of things. So while resistance to some of these therapies might be quite rare, what we really need to focus on is looking at other immune cells to try and come in with a secondary response to then continually target these tumour cells that might not be responding to the initial therapy. And a lot of what we've heard about tonight is really focused on T cell checkpoints. So that's with your anti-CTLA-4 and anti-PD-1. These are really focused on the T cells. So what my research is really focused on is trying to harness the, as they're called, natural killer cells. So they have an inherent ability to be able to kill tumour cells. And what we really want to do is use really basic biological research to understand how we can better harness this function that they have and also improve it. So.
Yeah, so we're far from done yet. In fact, I think, we've heard from a couple of the speakers earlier that, it started out that, a low percentage of patients were getting benefit, and then, as Peter said, 20% of all cancers at the moment get great responses. So that's not good enough, is it? I mean, we've got to do a lot better. So fantastic that people like Rebecca are taking up new approaches. So if you've got questions, please put your hand up and we'll get a microphone to you. I might turn now to our other person we haven't heard from tonight, Miles Prince. Now, Miles, you're pretty good at convincing people that they should invest resources in philanthropy in cancer. I'm going to ask you a question. So if you managed to get $100 million to invest in cancer research, where would you spend it?
Well, it's a good question, Grant. I mean, the first thing I would say is if you gave me $100 million, I would try and match it and try and find another $100 million because it's, look, it is a bit of a bottomless pit in terms of the need. There's always going to be a greater need. I mean, we have to keep researching. I think philanthropy actually is an unrecognised player in all of this. Sadly, a lot of people die from these diseases, and one of the fundamental immunotherapy we had for years was allogeneic transplant, where you get a bone marrow transplant from somebody else, your brother or sister, and that was the thing that made us realise that the immune system was important. Which is that if you get a donor, you're putting in a brand new immune system. For years, we just thought it was just the chemotherapy did it, but it's actually the immune system. But sadly, that can cause a lot of toxicities. And in fact, the Maddie Rewald vision, which is Nick Rewald's sister, sadly, she died from the effects of a bone marrow transplant. So the foundation I'm involved in, which was Snowdome, which links to the Maddie Rewald vision, we have no problems in speaking to people and saying, how can we support the new fundamental research? Because the goal is making hope real. And so people want to do that. They want to see their money being used valuably. And I think one of the things so the answer to your question is that I would love to see the government trying to support philanthropy, to match the dollars that philanthropy raises, because I think that's a way in which we can leverage a lot of money. But realistically, one of the things that we lack here in Australia is, I think,
a global vision. We rely on the health departments and we rely on our governments, the departments to drive the vision. We should actually be doing a lot like the United States, having a National Cancer Institute, which can actually drive a lot of these agendas and take these things forward rather than it being so reactive. So that's a long-winded answer to your question, except to say that I think we have a huge nidus of a vision here that is actually, you can see it in the audience, it's very easy to engage because there's just such enormous successes.
Yeah, I might, you've raised, the where to get the money from issue, so I might ask Alan Trousen sitting on your right. So Alan, now you've made fundamental discoveries in basic biology, in discovering and developing in vitro fertilisation, and turn that into commercial products that make it, a mainstay of treatment. So in cancer, are we doing enough in Australia to commercialise some of the amazing research, Rebecca's research that she's doing, and is going to have a great new application, hopefully, for natural killer cells? So are we good at commercialising in Australia, and are there better ways of doing it to what we're currently doing?
Look, I think Australia's pretty good at really funding some of the basic research. I think there's been a big investment, by government in the basic sciences, and I think Australia really has performed very, very well right across the board. But there is a problem of sort of making it connect to the patient. So when I was in California, I had $3 billion to play with for regenerative medicine and stem cells, and we created a revolution with that because we said, okay, if you make a discovery, we'll back you all the way through translation to the clinic. We won't stop. We won't say, look, if you're on track, and if you stay on track, and if you get there at each of those critical points, we'll keep backing you all the way. So, I mean, in some of those companies, we put 50-odd million, and they started off as an academic group, and they were converted at some point, probably to a company. Irv Weissman was here this morning and gave the example of CD47. Now, that's really powered up with two big tranches of 75 million, really from the investment, the financial sort of components of the US now. We could do the same here, and when I came back from California, I chose not to go back into university and go off with a begging bowl to NH and MRC for a few $100,000 that I might have got in competition. But I said, well, why don't we go out? I get Richard Boyd as an immunologist because if I'm going into cancer, I really need him. I need Miles Prince. I need a whole lot of people to join us in this endeavour. But if we can get money from industry to start backing us in a substantial way, and that if you get embedded in the research institutes as well, you have both industry, you have the possibility of getting government grants. We got a big government grant at the end of last year. So we're actually now starting to sort of make our way, and while we're relatively small, we've grown much faster than we could have ever done. If I'd gone back into one of the institutes and sort of put in my grants, it would never have worked that way. Richard would still be sort of wandering around in academia, very usefully, but he wouldn't be focused on this kind of thing. And our vision is specifically to take stem cells, bring it into the cancer area so that we can make an off-the-shelf product. Maybe that would be tenfold cheaper than having to treat the patients with their own T cells. That might make a really big difference. But as you said, Grant, it's going to take some time to deliver. And in the meantime, we'll probably do some autologous work to show that we're effective at what we're doing. And then hopefully we'll be able to have an off-the-shelf product that will be useful to the people who need it. But I think it's that combination of being able to work together, both in the research industry, across institutions, and say, look, we're not everything, but we could do with a bit of support here and there, and why don't we have a combination that will work? And that's the way I think it will probably progress more quickly.
So the idea of off-the-shelf T-cell therapy, very exciting because, Sharon talked about the checkpoints. Jared received the checkpoint inhibitors. They're off-the-shelf therapies. They can be given to any patient, but not everyone responds. And so the idea of being able to have an off-the-shelf T-cell therapy is pretty exciting. So Cassian Yee, off-the-shelf cell therapies, may be coming. Not quite there yet. Now, the B-cell leukemias, great results with this new cell therapy just approved last year, on the way now around the world. But B-cell cancer is pretty uncommon cancer, where we've got a lot of other huge problems. We've got lung cancer, we've got pancreatic cancer, breast cancer, bowel cancer. So what do you think the hope is for cell therapies for those, common cancers? What do we need to do?
I think first of all, the treatment for the B-cell leukemias was pioneered by basic research that dated back 3 or 4 decades, actually. And really understanding the mechanism in which you activate T-cell and get that to proliferate and respond in a way that can kill 1,000 tumor cells for a single T-cell has led to that evolution. So clearly, the CAR T-cell therapy for B-cell malignancies is a milestone in terms of not only raising awareness for the ability of these technologies to work in a clinically relevant manner, but also to sort of spur interest in the whole field as well. So it sort of has a dispersed effect under the umbrella of T-cell therapy. So now agencies are more likely to fund such research, philanthropy, and even the NIH, for example, which has been very slow to move in terms of high-impact, high-risk research. So, but moving from liquid tumors, as we say, B-cell malignancies, to solid tumors, and you might have noticed in the slide, I put these funny cells around the tumor, the stroma, those physical and also the soluble barriers to the T-cell getting in and doing its job. The solid tumor malignancies like lung cancer and GI cancer, colorectal cancer, they have their own set of unique problems that need to be addressed. So that really, I think we're at a level now where you've seen a steep trajectory of interest and success with CAR T-cell therapy B-cell malignancies. You now need a whole lot of innovation and discovery science to be translated to try and attack the tumor in many different ways so that the T-cells can get in there and do their job. This sort of idea of integrative medicine, the combination of immune checkpoints plus T-cells to allow the immune system. And if you're an immunologist, you sort of maybe bias into thinking all cancer therapies end up through the immune system. That may or may not be true, but you really have to be careful now which drugs you use so it doesn't handicap the immune system from doing its job and which one's promoted. But I think this is really the next sort of major obstacle to overcome is how do we get past these barriers which a tumor has been smart enough to co-opt to its advantage.
So I might turn to Sharon Lewin. So you lead an institute that focuses on both infection and immunity. And there's been some really interesting research emerging about the microbiome and the bacteria, particularly in our bowel, and how that might interface with a whole lot of different immunologic-based conditions. We have antibiotics that are widely used, and for a lot of even poor indications. Is this area a real breakthrough, Sharon? Do you think for the treatment of immune-based disease, or is it just a bit of hype?
I used to think it was a bit of hype, but I'm totally converted to the opposite. So what Grant's getting at is the microbiome is basically the germs that live in our gut, or actually live in other parts of our body, in our lungs and the genital tract. All of us are filled with all these different germs that we've never really thought do much, but we're now discovering that these germs, or the germ profile, is very strongly linked to certain diseases. And in this particular, and actually a whole range of different diseases are linked to the microbiome. And the microbiome, as in what all of your bacteria look like, is heavily influenced by your diet, by how much antibiotics you've been exposed to, by what antibiotics your mother might have been exposed to at the time of delivery. Quite incredible imprints on our microbiome. And the most interesting association for me was the link between what your microbiome looks like and how you respond to immunotherapy. It's quite incredible. We haven't taught much about how to predict who responds to immunotherapy. So maybe 50% of people will respond, but at the moment we can't predict who will respond. And there was this incredible research done that if you had a certain type of bacterial profile in your gut, you had a greater chance of response, and those differences were marked. So this is really fascinating. Can we take advantage of altering the microbiome to improve your response to immunotherapy? What does that mean about how we prescribe antibiotics? And just one last quick point. We sort of antimicrobial resistance, or superbugs that are resistant to all the antibiotics we have, is now recognised by all health authorities as the next great challenge in infectious diseases. When you've got someone sitting in front of you and you say, I don't, you don't really need antibiotics, but perhaps we'll do it just to be sure, and it's not good to use antibiotics because it spreads resistance. Most people want that antibiotic because they're just thinking about the chance that they might be sick. But if you know that your antibiotic that antibiotic will irreversibly affect your immune system in the future, I think that will really change the way people think about antibiotics. So it's a really exciting, very, very new area of research that may have big implications for how we can get immunotherapies to work better.
Yeah, thanks, Sharon. And we've got a question, so if we get the microphone there. I might just say, Sharon, but while we're waiting for the question, so Sharon mentioned about trying to predict response to these immune treatments. So, Jared, you actually had some T cells inside your tumour that we cut out, which we were pretty positive about because that's a predictor, and the lumps were pretty small, but it still was a bit of a roll of the dice. We weren't sure, were we?
No, not at all.
Yeah. So we do need these new markers. Please, question.
Hi, I am a Carlton supporter. Please don't hold that against me. But my mum is a mad keen Hawthorn supporter, so I am winning Daughter of the Year. Yeah. Thank you so much for that opportunity. I am a research scientist, and from a patient's perspective, it would be really interesting to know if you wanted to know more about the science, or if, clearly you've got a great relationship with your oncologist. Do you, as a patient, do you care about the science at all, or do you just want to be fixed? But thank you for being here too.
It's obviously nice knowing what's behind it all. It's easy just to say, here, have this, it'll make you better. But knowing what effort has gone into it, what can and can't happen in terms of the side effects, and be prepared and whatnot. I guess I was as much of a blank canvas for these guys, and it was vice versa, because I was in daily contact with Grant and Donna saying, this has changed, this is the same. So I had to be pretty open with them too, because I guess I was one big cell in a sense. They're just working on me.
And Jared, in terms of the importance of scientists, when you were having your plasmapheresis, you met a bloke with a beard came to visit you. Do you remember?
Yeah.
Jim Allison, right?
Yeah.
And Jim, Peter spoke about earlier and showed his photo. So how important do blokes like Jim Allison discovered the checkpoint that Sharon spoke about? How important is he to you, and do you think other patients?
Well, I'm only here because of him, effectively. So I was pretty happy to meet him when he told me. Well, you said he'd won a Nobel Prize as well.
Not quite yet, but Peter, really?
Mate, as well. I'll give him my vote.
Peter, by the way, has written a book on how to win a Nobel Prize. Great, we've got a question here. I think we'll take, this may be our last question unless there's one more pressing one.
Thanks, Grant. Graham Down, I was diagnosed as having stage 4 melanoma in November 2011. So as I said to somebody the other day, if you want to be in the wrong place, be there at the right time, where my experience has been. I should have been gone a couple of years ago, but thankfully for the research that's been done has been absolutely gobsmacking for me. So I'm not only still here, but I can actually ask a half-intelligent question. The last 12 months, I was gobsmacked at your presentation that you gave with Jared. I was gobsmacked that, Jared, you only had 3 treatments of Nivo. I started on pembrolizumab 12 months ago, and I've been having it every 3 weeks for 12 months, whatever that adds up to, but it's a hell of a lot more than the 3 you had.
Yeah.
And I've had a couple of nasty little side effects along the way, but they're livable. It's early days, I know, but the question's largely to you, I think, Grant, is how long does this go on for? Obviously, you had 3 doses with Jared, and that was adequate sufficiency, and he's back playing football. Goodness me, it's not just a kick in the park either. He's a very, very good footballer at the most elite of levels. How does he have 3 treatments and walk back into AFL football? Where are we up to? As I said, I'm 12 months in, and as my oncologist said to me, if it ain't broke, don't fix it. If your side effects are manageable, we'll just keep giving you the dope. So my guess is that it's...
Great question, Graham. Thanks for sharing that. So Jared, of course, as we discussed, had to stop because of side effects. And it's interesting, actually, there is some association between side effects and how well the treatment works. And so maybe, if one gets side effects, it can be a sign that you don't need treatment for as long. But it's a fundamental question we don't know the answer to. There are trials planned, stopping trials. So stop the Opdivo or the Keytruda in half the patients and see if there's any relapse. Those trials are planned. But I might, because it really is all about the science, I might ask Cassian Yee. Cassian, how long do you reckon we need to keep these, what's your guess, these PD-1 treatments? I mean, you've spent your whole career studying these T cells. How long do we need to keep these treatments going for?
Not to affect the stock market of these drugs in any way, but I think that, just to back up a little bit, there are 2 immune checkpoint inhibitors that were presented. One was the anti-CTLA-4, which Jim Allison helped pioneer. He just works down the hall from me. And the other is the PD-1 therapy, the pembrolizumab or Nivo or Opdivo or whatever you want to call it, which you've had and which can be given every 2 or 3 weeks. I can tell you, honestly, we don't know. Although we've had patients who stopped after a year or 6 months, either because of toxicity or follow-up, and have had long-lasting responses. Now, there is science behind this, and that is, is the PD-1 acting at a different stage of the cancer-fighting T cell, and the anti-CTLA-4 is acting at an earlier stage in which you can generate a broader response that lasts longer. So anti-CTLA-4 is only 4 doses every 3 weeks. However, the studies show that PD-1 works for many more types of tumor than anti-CTLA-4 has. Anti-CTLA-4 has more toxicity. So there is actually science between a drug that's given for short term and a drug that's given over the long term. And some of the studies actually our lab has done and Jim has done has shown that PD-1 reactivates cells in the tumor site and requires that blockade of PD-1 for them to remain active. Okay, and that's a little bit different from the other drug. I think that this is something that for the research requires perhaps even tumor biopsies to see what's going on at that time. But it's really, because we have a wealth of drugs now, not only PD-1 and anti-CTLA-4, but some of the other drugs, I think, that were shown earlier, maybe these combinations will allow you to shorten the period of therapy. I'm sorry, that's the best answer I can give.
Yeah, and the need for more research. Can we all give Graham a round of applause? 2011. And actually, I think that's a great, note to finish on. Is just remarkable, outcome for Graham of having the treatment. And so, it just remains me to thank all of you for attending this evening. Our panellists have been absolutely fantastic. We've got some great stars here, both locally and internationally, world leaders. And especially thanks to Jared for sharing his personal story, and made all the best for the Hawks for 2018.
Thank you.
Yeah, thank you.
Well, I don't know about you, but that was just extraordinary. I've been working in immunology for 40 years and I'm nowhere near going to get a Nobel Prize. But when I finished my PhD, my wife and I went to Austria, Innsbruck, and it was a wonderful place to work. But coming from Australia, they thought that I was working on immunology rather than immunology. So it's something rewarding to see that immunology is having its moment in the sun. And I think, as Grant summed it up, it's an extraordinary event to have the doyens such as Peter Doherty, a living national, international legend. He can go home tonight and say, gee, I did a pretty good job, didn't I, really? He can relax, but he doesn't relax. He's always at it. You ring him up and he'll give you 15 minutes of high-powered science and set the scene beautifully tonight. And Sharon and Cassian, again, filled us in with the basic science which underpins these discoveries. And from them we move into Jared, the extraordinary story of Jared Ruffin. Not only is he a fantastic footballer, but what a great guy, you know, from the country. But you sort of said that like an excuse. I think it's just a badge of honour. And you presented your story. You're an inspiration to all of us. If you can convey your message as well as you do, it gives everybody in this audience a sense of, you know, there is some real hope, and we should get away from the word of fear. But what we do learn is that for every success, we've had a couple of brilliant ones today, there are failures. And so now is not the time to bask in the glory. It's a time to, I think in one of Sharon's slides, let's put the foot on the accelerator and drive this immunotherapy revolution to, if not eradicating cancer, then at least converting it to a manageable disease and not die from cancer. So I'd also like, as Grant, the doyen of actually he's probably the Gerard Whateley of this sport, really. He knows every statistic and he's a fantastic doctor and a fantastic guy, and I'd like you to join me and thank all our speakers and panellists one more time. And so, even though the liver's fixed now, Jared, so there's some grape-flavoured tissue culture fluid that you can use to build up some resistance. So thanks once again, and thanks very much to all the members of the general public. You play an important role. You're the people who can drive the governments to help bright young scientists like Rebecca and her fellow colleagues to be the superstars of tomorrow. And thank you very much once again.